Welcome to Who's in STEM. I'm Ken Ono, your host and the STEM advisor to the Provost and the Marvin Rosenblum Professor of Mathematics at UVA. Our goal is to evoke flights of imagination and wonder by showcasing the cornucopia of all that is STEM at UVA. The marvelous world of UVA science, technology, engineering, and mathematics. Now, we've talked a lot recently about artificial intelligence, its impact on universities, its role in scientific discovery, and the need for guardrails due to potential threats and future risks of AI and AGI, known as artificial general intelligence. Now, AI evolved from decades of research by computer scientists and mathematicians who mimic brain neural networks. And what they discovered is that they could mathematically model the intricate and complex mesh of synaptic connections that seem to be essential for higher brain functions. Today, instead of discussing artificial intelligence, I want to focus on actual brains. At UVA, we have an exceptional team of scientists thoroughly investigating the brain. Our brains are delicate, vulnerable to disease. You might remember Jaideep Kapoor talking about epilepsy and Sarah Kasinis talking about brain development. Also, on other episodes of Who's in STEM, we've talked to George Bloom and John Lukens, and we've heard about their contributions to Alzheimer's research. But these aren't the only conditions that concern us. I know firsthand, 15 years ago, my wife Erica had a brain tumor, discovered after a grand mal seizure, and I'll tell you, that's a frightening thing to watch. The very next day, she was on the operating table, and surgeons at the University of Wisconsin removed a golf ball-sized meningioma from a sensitive area of her brain. And thanks to modern medicine, she's fully recovered, with only a depression, a divot in her skull as evidence. So I'm very grateful for the work that leading neuroscientists and medical researchers have produced over the years. Today, we welcome UVA neuroscience star and brain cancer researcher and also chair of the neuroscience department, Professor Harald Sontheimer. He's a leading figure, and it's a pleasure to have you on Who's in STEM.
Well, Ken, I'm happy to be here. It's always fun to share my passion for science with a scientifically minded audience.
So, Harald, there's a lot to discuss, but I want to begin with huge news. You have received the 2025 International Prize in Translational Neuroscience, one of the top awards in your field. For a layman like me, can you tell us about the work, the award, and what it all means to you?
The past 25 years, the German Max Planck Society, which was one of the largest funders of science in Germany, has been awarding a prize in translational neuroscience to recognize scientists who made seminal contributions to this field. This is a field that bridges fundamental neuroscience and clinical applications, and each year they pick a different emphasis area. This year, they recognized cancer neuroscience, a relatively new field for which I'm one of the pioneers, and I'm humbled to be sharing this award with my colleague Michel Monnier from Stanford University. Now, neuroscience has been around for 100 years or more. So has cancer biology, has been around for at least 100 years or so. But we're really looking at a fundamental pivot in terms of how these two fields interact with each other. Traditionally, brain tumor or glioma research has taken a chapter from other cancer research. So they've looked at colon cancer, prostate cancer. What are the shared genetic mutations? What are the shared features, signaling features that drive aberrant cell proliferation? And essentially look at gliomas as just another cancer that so happens to grow in the brain. Now, my laboratory, beginning 30 years ago or so, has taken a very different tact and has asked, so this is a tumor that starts in the brain, it grows in the brain, it stays in the brain, and it arises from brain cells.
The pathology really is different?
Yes, it really is different. For example, we'll come back to that later. You'll never hear of a brain tumor metastasizing to the prostate. But your prostate cancer could metastasize to the brain. And we're going to get into why that is, and that's fundamental to this sort of cancer neuroscience field. So I called my approach at the time a neurocentric perspective, where instead of looking at other cancer, I took cues from how are these cells different from normal neurons, from normal glial cells and stem cells from which they derive? So normal glial cells provide a hospital environment for the brain. They essentially regulate ion composition, they deliver nutrients, they regulate the blood supply to the brain, but they don't typically signal in the classical sense, like they don't form so-called action potentials to signal with nerve impulses to other cells. But it turns out that when they become cancerous, many of these supportive roles that these glial cells play are lost, and instead they acquire some of the phenotypes of bona fide nerve cells, and one of those is ion channels. We're going to come back to how I got to study ion channels in these cells in the first place, but it was interesting to find that the same ion channels that are used in neurons to signal to each other, namely sodium, potassium, and chloride channels, are actually utilized or expressed by these glial-derived tumor cells. And we were initially bewildered by this finding. But we quickly found that these ion channels were repurposed for different functions. Cancers that form metastases typically do so by getting in the bloodstream. And so there's some randomness to that. And I pointed out to you that gliomas don't do that. So one of our early questions was, why is that? And to answer this question, what we did is we took mice in which we implanted human gliomas. So these mice then recapitulate the salient biology of the disease. We then took a piece of the skull off the mouse and replaced it with a glass slide. We then can take this mouse under anesthesia, under a laser scanning high-resolution microscope that allows us to image deep inside the brain to find these cancer cells. So what we're finding is that they're sitting on the outside of the blood vessels. They're not inside the blood vessels, they're on the outside of the blood vessel. In fact, they're lined up in a string, moving along the blood vessel, using the blood vessel as a highway for their migration. As they're going along, they're having to mold themselves into the narrow spaces that are left around blood vessels, between the blood vessels and the neurons and glial cells of the brain. So that gave us a clue as to why these cells might express ion channels. What we found is for the cells to mold themselves into this space, they have to change their cell volume. They have to shrink and increase in volume again. But for a cell to shrink, you release ions across the membrane and water will osmotically follow. It's almost like, in your sweat glands, when you want to secrete fluid, you secrete salt, and the water will follow. So we found that they're utilizing potassium and chloride channels that nerve cells use to signal to secrete salt with cytoplasmic water to follow, allowing them to squeeze through these tiny, narrow spaces. Mm-hmm. So they've repurposed them in a way to allow them to invade the brain. And it raised the spectra, of course, for these ion channels as being the gates allowing these cells to invade for being a potential therapeutic target.
So the tumors that you are studying, as you've described them, presumably then form shapes that are rather chaotic, right? They don't necessarily form tumors with well-defined margins. It must look like a Jackson Pollock, right?
They very much are. I should mention that these studies revealing ion channels in gliomas, as well as normal glia that we compared them to, were among the first to show that non-excitable cells express ion channels, and particular cancer cells. And many studies have followed on and have now shown that ion channels are actually used in all kinds of organisms, including plant cells. And there's now a cottage industry studying ion channels in cancer, with a number of drugs that target ion channels actually being in clinical trials. So very happy to have been among the first to actually do that kind of work.
So you won the award for this discovery that has opened up this entire field of research on cancer, as well as providing approaches for drug development.
So the translational aspect is important. I was initially at Yale and then at the University of Alabama, Birmingham for 20 years. And it was really during that time that we made some of these seminal discoveries. And having identified channels that could potentially be blocked and thereby inhibiting invasion, we did a lot of basic science studies showing that indeed in a mouse model of the disease, we could stop the tumors from invading. And then we went on a year-long search for drugs that could inhibit this ion channel, or one of these ion channels. And we initially honed in on the chloride channel because it was highly expressed in the gliomas, but not really much expressed in other brain cells, so it could be targeted effectively. And then we went through just about all the available potential drugs that could block these channels. And it took a long time because these studies are very tedious. And we arrived on a scorpion toxin called chlorotoxin now.
So actual toxin that is...
Actually toxin from scorpions. Now, for any ion channel biophysicist, no one would raise their eyebrow because it's a characteristic feature almost of all of the ion channels, particularly potassium channels and chloride channels, to be blocked by poisons from either scorpions, poisonous spiders. The sodium channel is blocked by tetrodotoxin, the pufferfish toxin. So there has been this coevolution of predator and prey because often these channels are used, either in muscles to contract or in the nervous system to control movement. And the predators simply block these channels to paralyze their prey. So there has been this coevolution. And so it's not surprising to those of us who study ion channels to find that in the scorpion. Now, of course, what do you do with the scorpion toxin? You're not going to milk scorpions and give that to patients. So I started a biotech company, Transmolecular, in 1995, and we started doing not just these drug screens, but creating a synthetic form of this molecule. It was a very difficult endeavor because the molecule would just never form in the right three-dimensional way to stick to the channel. But ultimately, we succeeded in doing that, in doing biosafety studies, stability studies, and introduced that into mice and ultimately cleared the way to do clinical studies in humans. Mm-hmm. And in fact, we were able in our phase 1 study to treat 12 patients, and the phase 1 study is simply a safety study to show that we don't do any harm, and these patients were perfectly fine receiving the drug. And then we went on with 59 patients to do a phase 2 efficacy study, and the results were actually quite stunning. We tripled on average life expectancy and had about 20% of patients being long-term survivors from this. And so that was very exciting. And then the FDA cleared us to do a phase 3 study in 15 centers around the country.
And when was this?
This was in 2008. Mm-hmm. And some of you may remember that the markets crashed.
Yeah.
And it took us down. So getting to this point, we had $42 million of venture investment. And now we needed about $60 million to do a 15-center open label trial. And the FDA actually had designated this as a pivotal trial, meaning that at any point in time, if the results continue to be as positive, they would simply stop and go to market because there is no treatment for these cancers.
Oh, I didn't know about that. So for listeners, in drug development, there are generally 3 trials, phase 1, phase 2, and phase 3. And for diseases where there are no options, phase 3 can be curtailed or fast-tracked to...
That's right. You can essentially break the blind at a certain point and say, this looks really promising. There's no need to do another 200 patients and do placebo control and whatnot. We can go straight to market because these patients are
lives are at risk, right?
Given 4 months to live in hospice care. So you don't want to do that. But unfortunately, we couldn't raise the capital. And the investors that had invested in the company up to that point were getting increasingly impatient and eventually sold the intellectual property to a big pharmaceutical giant, Isaiah Corporation. While it was sad to see that happening, ultimately it's gratifying to see that the molecule really hasn't died. There is really interesting clinical studies going on using a fluorescent form of this molecule as so-called tumor paint by a company called Blaze Pharmaceutical and a colleague Jim Olson at the Hutch in Seattle has been doing that. They've recently developed a molecule where they take the sclerotoxin and conjugate it to the body's own immune cells, the so-called CAR-T T cells. Mm-hmm. So you can make the T cells recognize, a foreign epitope. And in this instance, they're directing these T cells to bind to gliomas. So I think there's hope that in the next years or so that this finding that we made on chlorotoxin really will get a new life as an immunotherapy.
So is chlorotoxin still in an ongoing phase 3?
So the company has never taken that phase 3 trial on. So drug companies often purchase interesting intellectual property or interesting technology only to change their mind. Some companies change direction. They go from cancer to Alzheimer to Parkinson. And so, I think in this instance, this very promising drug really became the victim of a market crash.
What do you predict for the future of chlorotoxin now that the work is ongoing? Do you have a prediction or a hope for the millions that are struggling with brain tumors?
I do very much, and I think it really rests to a great degree on the interest of people in academia. One of the issues we haven't touched on is the fact that glioma is an orphan disease. An orphan disease is a disease that's not very abundant, not very popular, and for which there's no treatment. But from a cancer perspective, some people would call it a boutique cancer. There's only about 100,000 cases a year.
That's a lot, if you ask me.
It's a small but for a drug company to develop a drug, you look at your market size, and what are the big markets right now? I mean, you look
Alzheimer's
and appetite suppression, and diabetes, chronic diseases, and yes, so even among the cancers, this pales compared to breast cancer or lung cancer. And so I don't think that this would be the primary market for a drug company. So it either takes a smaller biotech company or academia to drive that forward. And that's why I'm very excited about the idea to conjugate this with immune therapy. And that typically happens in academic center, those kinds of approaches.
It is unsettling that economics influence decision-making in science as you've described it. It's troubling. Presumably, tens of thousands have lost their lives over the last 10 years because of this delay in the research. I think here at UVA, there are reasons for optimism. A key goal of UVA's Manning Institute. Just the other day, I was in Fontaine Park, and it's exciting to see this building pop up out of the ground. One of the key goals of the Manning Institute is to make sure that research doesn't fall through the cracks, either because of problems with regulation or lack of funding. And earlier this summer, we had Mark Esser, the first chief scientific officer of the Manning Institute, on this episode, and he talked quite a bit about that. So your work at Transmolecular, your startup, aligns well, I think, with UVA's objectives, and I hope that the future helps this major breakthrough become translational, make it so that it helps the lives of actual people, because what good is science if it's not helping the human condition?
It's actually, I would go further. Had the Manning Institute been there when we made this discovery, we wouldn't have needed the company. So really what we were struggling with is, how do you make a synthetic drug? Well, there was medicinal chemistry, and NanoStar as a delivery platform right at the Manning Institute. They have to make your molecule to good manufacturing standards, good manufacturing practice, so you need a GMP facility. It's right in the Manning Institute. Right. So you can do the development of the molecule and the translation in-house. And that would have probably saved us not just millions of dollars, but the whole headache of fundraising.
It would have facilitated the whole process. So I know your portfolio is quite broad. So can you tell us a bit about other ongoing projects in your lab?
Well, the second major project related to cancer neuroscience, and that pertains to that award, that's really occupied my lab for the last 25 years, focused on the neurotransmitter glutamate. Now, glutamate is the language used by neurons to talk to each other. One cell releases it, the other one responds to it. There's specific receptors for that. Too little of it, you have memory loss. Too much of it, you suffer seizures. The Goldilocks concentration of this molecule is achieved by regulation by glia, specifically astrocytes. They take up any extra glutamate that's being released from neurons and converted to glutamate, which makes it harmless, and it serves as a precursor for the new synthesis of glutamate by neurons. Again, when we started comparing normal glia, normal astrocytes, to these glial tumors, gliomas, we found that this ability to regulate glutamate and contain it in the brain at non-toxic concentrations was lost when they became cancerous. Not only that, the tumor would actually spit out extra glutamate to concentrations that made it toxic to neurons. One of the reasons most glioma patients present with seizures as a presenting symptom is there's an overabundance of this glutamate being thrown at them from tumors, make them hyperexcitable, and a few days later they succumb to their hyperexcitability. So the question became, why in the world would a system like that evolve, if you will?
Yeah, what is the biological benefit?
The benefit. So think about it. Gliomas are the only cancer that grow in a completely enclosed space. It's your skull. Any other cancer grows against soft tissue. They can grow the size of a baseball, whatever, in the colon, in the breast. In the brain, not so much. So for every inch of space that the tumor takes up, an inch of brain has to go. So the tumor actually has to destroy the brain. And I would argue it's probably the most vicious tumor in this regard. It's probably the only cancer that actually destroys the host organ that it grows in. And the culprit for that is glutamate. So we spent 2 decades figuring out how is the glutamate made, how is it being released, can we stop it, how does it contribute to the growth of the tumors. And very excitingly, we found that there is a amino acid transporter. A transporter, unlike an ion channel, usually moves a substrate in one direction with another substrate in the other. So in this case, it actually is a molecule like a revolving door that releases glutamate going out for cysteine, another amino acid going in. The cysteine is one of the essential building blocks of glutathione, an antioxidant, highly expressed in metabolically active cells. And, some of your listeners might actually take supplements of glutathione to treat oxidative stress conditions or muscle stress.
Yeah, I think I've seen those ads at 3 a.m. on cable television.
So it turns out from the cancer's perspective, you simply have a cell that's metabolically active. It needs a lot of this cellular antioxidant. But in terms of importing this building block cysteine, it releases a lot of glutamate. And
Can you interrupt that?
Right. So, and we embarked this time without a company in the lab on a chemical screen, and we identified a number of molecules or chemicals that can block this transporter. Among those, we found a drug that's already FDA approved for a different condition. The drug's name is sulfasalazine. It's used by millions of patients that have Crohn's disease. It latches onto the
gastrointestinal.
Exactly.
Yeah.
So it latches onto the transporter, and it's too big to be transported, so it jams it up. It's not a good drug in terms of its blocking ability. You need high concentrations. It has poor bioavailability. But it works, and it's safe. Patients take gram quantities every day. So it allowed us to do a clinical study to simply ask, can one inhibit glutamate release from tumors? And if so, does that actually change seizures or hyperexcitability? And we've been able to show that, and show that, in fact, this transporter is the culprit that releases the glutamate. And the only reason sulfasalazine isn't in clinical study as a treatment is because you would have to take it almost every 2 hours, because it only sustains for a very short period of time. So there's currently a number of drug companies and academic labs trying to develop similar molecules that bind to the same site but have much higher affinity and a longer lifetime in the body, so that one can use that as a long-term therapeutic. But we're very excited about that finding, and it certainly, again, links back to this idea of neuroscience in cancer, because here's a bona fide neurotransmitter and the cancer takes advantage of the signaling between nerve cells to overstimulate that pathway to vacate space by killing off these neurons, by vacating space for the tumor to expand, and then for us finding a way to inhibit that pathway is very exciting.
So I'm very curious, as a scientist, you discover that an FDA-approved drug, sulfasalazine, offers temporary, very short-lived benefits in disrupting this glutamate process. So as a scientist, what do you look for next? You said you would look for a replacement. How does that work? Is that part synthetic chemistry? Is it searching around for other drugs that perhaps had been proposed treatments for Crohn's? I mean, how does that work?
Yeah, so there's multiple strategies to use. First, you have to identify how the sulfasalazine blocks. And so we found that it actually, one part of the molecule mimics the molecule glutamate that's normally transported. So it docks onto the same site where normally the glutamate docks on, but it has a longer tail hanging off, and therefore it jams up the revolving door, if you will. So now you could say, okay, let's look for molecules, use a medicinal chemist or chemical engineer, look at, can we find that moiety that's similar to glutamate but isn't glutamate in other chemicals that are already synthesized? And if not, can we synthesize something similar to the molecule sulfasalazine? That's one strategy.
Is that what you do? Do you partner with chemists?
Yeah, absolutely.
Yeah, okay.
And I think that's where the Paul Manning Institute, again, comes in. You have medicinal chemistry there, so you have it on site. You can discuss these with people. The other one is, science is collective wisdom. Science is a lot of people working, and someone who doesn't study glioma might be interested in this protein for a completely different reason and accidentally discovers a molecule that binds to it and blocks it. So you test that out and see, well, can we hone in on a different molecule to do that? And sometimes it's just dumb luck, that you stumble onto a drug that actually works. But inevitably, a good scientific study ends by saying we've identified a novel target. A target is different from a treatment. It basically means that here's a channel or transporter that could be exploited as a treatment, but finding a drug for this target is often awfully hard and sometimes impossible. Now, we've identified a whole family of very high affinity, very potent blockers of this transporter. They're called phenylglycines, these drugs. But they also bind to other glutamate receptors on neurons in the brain. So they have very severe side effects. They could never be used, for the treatment of glioma. So you have to balance, the perfect high affinity drug versus specificity for your target.
Now, I want to go in a slightly different direction now, going a little bit beyond the research. I understand from many, including Lori McMahon, our Vice President of Research, that you have had a significant impact everywhere you've been in terms of institution building, and we like that at the University of Virginia. Can you speak to that? Can you tell us a little bit about the work you've done as an institution builder?
Well, I had a couple of fortunate opportunities given. One was when I was at UAB. And so they gave me an opportunity to really marry my interest in glia and glial cancers, but also in general the role of glia in disease processes to create the first Center for Glial Biology in Medicine. So an organized structure where I was able to hire like-minded people and to really build up a whole cohort of people that were looking at glial cells as the solution for neurological illnesses. We had annual conferences and whatnot. But what drove me away from UAB, or what pulled me away, was Virginia Tech asked me to consider building a school of neuroscience. And that's probably the biggest endeavor I built from the ground up. We built basically a undergraduate directed program where we administered 4 majors in neuroscience: experimental, clinical, computational, and cognitive neuroscience. Attracted 1,000 students, became one of the most popular majors in the College of Science, has now been in existence since 2015. That's when I moved there. I had to go through all the state approvals to get not just the majors, chef, and all of these things. But really the challenge and the opportunity was to re-envision what an education in neuroscience should look like. Rather than taking a physiology department and a biology department, getting together and figuring this out, basically hiring 25 neuroscientists, building a curriculum from scratch with really cool courses, War in the Brain, Global Perspectives in Neuroscience and Medicine, I mean, things like that, to neuroscience in society, and really creating a magnet program for students who want to study neuroscience. I'm probably most proud of that. And then coming to UVA, it was just great to take over leadership of a department that is so uniquely known for its emphasis on neuroimmunology, a real strength here, and really having been at the forefront of that field and being a leader there is very gratifying.
So Harald, I have to ask, it's not every day that a world-class researcher is also an institution builder who had to think quite deeply about assembling a curriculum from scratch. I mean, that's quite a portfolio. From a very broad perspective, I want to ask, share with us your views on academia, mentoring, and education, all of the above, because your experience and success is quite unique.
Well, foremost, I'm a professor, as are you. I'm not a researcher. As a professor, I see my role as a teacher, teaching the next generation of learners at any level how to do fine science, how to formulate a hypothesis, how to test it, how to communicate it, how to publish, how to tell your story. And the science that you do become a vehicle to train students. Now, if you think through that for a moment, it slows you down in your endeavor, but you actually have a shared mission, not just to do excellence in research, but to actually bring in new people into this field. And you take rewards in a different way. It's not about having the coolest science, but actually seeing those people go into the world and live their dream and make you proud and publish way nicer work than you've ever published.
Right. It's actually the 10x factor through your legacy.
Yeah. But one thing that we often overlook is that these young minds are actually broadening your mind. So I would argue that we do better science in universities where we involve trainees than we do in research institutes, because we have these fearless young minds that have crazy ideas, and a lot of these crazy ideas actually work.
Yeah, they challenge you.
They challenge you, yeah. Right. So I would credit all our discoveries in my lab. I would credit all my trainees for that. They've worked hard, and they came up with really fun and crazy ideas that actually worked. And so that's one of the motivations to keep building institutions that are built around learning and knowledge. Now, when it comes to what a curriculum in neuroscience looked like and how did I have the capacity to do that, well, you could also say, how did I have the arrogance to propose one, because the way that story went is I actually started teaching a flagship course called Diseases of the Nervous System.
At Virginia Tech?
I actually started teaching that at Yale and then at UAB, and it became increasingly popular. I had typically MD, PhD, med students, residents in there, but at some point I had the honors neuroscience students in there, and they kept begging and bleeding that there should be a book for this course. And after years and years of hearing this, they wore me down and I said, I'm going to write it. So I started writing a monograph.
The book is called
Diseases of the Nervous System, Elsevier, publisher. It's in its second edition. Won the Prose Award of Best Textbooks in 2016. So was really happy about that as a novice book writer. But it was very gratifying because I was able to read thousands of articles and distill them down to the level of an undergraduate. And because of that experience, being really immersed with undergraduates, with undergraduate learners, and reading all these articles to come up with just how broad and informative neuroscience is, I called it at some point the new English major. You know, if you don't know what you want to do with your life in the past, your mother would have told you pursue an English major. I would say today pursue a neuroscience major because it teaches you a lot of the, the important things about data, about perception, about how mood influences, your decisions and all kinds of things that are very useful. Anyway, so based on that experience, I felt in a very good position to dream up an ideal curriculum. And of course, I hired faculty and they infused their knowledge into this, and it was fun.
At the end of the day, I think all of us who are on the faculty share this love of our individual fields, but certainly have benefited and enjoyed the process of mentoring the next generation. So speaking of your role as a mentor, let's wind back the clock. Someone presumably mentored you. How did you get started in your research?
Well, I actually had a really interesting first several years of my research career. I got my undergraduate degree in physics and biology.
In Germany?
In Germany. I was curious, how could I apply physics to living beings or biology? I was very fortunate to run into a professor who was a biophysicist studying comparative evolutionary neurobiology. And what that is, it's you look at organisms across the evolutionary spectrum.
So like zebrafish versus primates.
Absolutely. Yeah. Or even further, you go to amphibians, or you even go to, multicellular worms, which are jellyfish. But anyway, primarily focused at the later end vertebrates and look at how has function changed as structure changed, or how did the structure need to evolve to support function. And I was able to study eye movements.
As an undergraduate?
I was doing a master's degree.
Degree.
In Germany, you had to do a master's degree, an experimental master's degree for 2 years before you could enter a PhD program. So I joined Peter Hoffman's lab, and he was a vision scientist looking in an evolutionary way at eye movements. And the whole idea was that with further evolution, we get more and more binocular vision. Now, for our vision to be stable in a moving world, we have reflexes that constantly reposition our eyes as the environment moves. They're called the nystagmus. And it turns out that all vertebrate animals have a nystagmus that stabilizes the field. Like if you're a horse running down the prairie, the field would move from the nose to the ear, and the eyes need to jump back towards the nose and stabilize the field. It turns out that once you become binocular and you have a big binocular vision field, you also have to stabilize the movement of your head, voluntary movements, as now your environment moves, and it moves the other direction. So you needed a second nystagmus that stabilizes the opposite direction. The hypothesis was that this is a cortical function, and it involves both projections from both eyes, and that it's mediated by the corpus callosum, the structure that connects those two brains. So I got involved in this research and actually showing that, yes, when you cut that corpus callosum, this is gone. And yes, when you study an animal that doesn't have the corpus callosum, marsupials, for example, kangaroos, or I used a certain species of gerbil that doesn't have that particular optokinetic reflex is not there. So that was very, very exciting. My PhD work coincided with Irving Nair and Bert Sackmann, who earned the Nobel Prize in 1991, having just published their methodology to record single channels using the patch clamp. Now, what that is, you can essentially monitor the conformational change of a protein in the membrane, the protein wiggling just a little bit by like 2 or 3 angstroms, and by wiggling, opening a gate, allowing ions to cross the membrane. So it's basically the most elementary, almost atomic type of activity in the nervous system. And I just needed to study that. I just needed to go from a whole brain here to a single molecule in the membrane. And I searched high and low, and I found a young scientist in Heidelberg, Helmut Kettenmann, who took me in and allowed me to build this technology And he happened to study glial cells, the cells that I would study the rest of my life. At the time, I didn't care where these channels were expressed, whether they're in nerve cells, in muscles, or in this instance in glia. I just wanted to use this technology and study these channels. And so by serendipity, I put the electrode, I was probably the first to put an electrode on a glial cell, finding ion channels in non-excitable glial cells. And we spent several years studying them and having a great deal of fun doing that. Along the way, I was the only person at some point in Heidelberg, where I did my PhD, who at the time was able to do these kinds of recordings. So as some of the colleagues started cloning the first neurotransmitter receptors, which are ligand-gated ion channels, I was the first to put them into cells and record, for example, the benzodiazepine binding site or Valium binding site on the human GABA A receptor. So it was just really focused on studying neuroscience at the single protein level, and that was very, very exciting.
So can you briefly describe the technology, right? So what
Yes.
you're describing at this scale seems impossible.
Yeah.
Yes.
So you basically take a fine glass needle and you heat it up and you pull it out with a machine in a linear fashion so that the very tip is 1/700th the diameter of a human hair, about a micrometer. Now you push that very gently on the surface of a cell.
So without destroying the cell.
Without destroying
right.
this little piece of membrane that's only 1 micrometer in diameter. And in a good situation, there's just one single ion channel under this patch of membrane. With an amplifier, you can amplify the signal tens of thousand-folds, and now you can see as a quantum unit, when the channel opens, ions fluxing through, and as the channel closes, you see this, they're like square-shaped events. And you can see basically this molecule flicker as it opens and closes. And then you can change the voltage across the membrane, so you can modulate its opening and its closing, and you can characterize this function of this protein, at this sort of anatomic level. It's really fascinating.
What did it feel like when you first conducted these experiments and did this modulation? How powerful did you feel?
I never felt powerful about doing experiments. I just felt fascinated, in awe. It's one of those things you can't leave the lab. You've got a successful recording. You'll just stay there however long it takes. It was just fun. And I couldn't get enough of it. I actually, together with my brother, who was an engineer at the time, he became a physician later, but we built some robots that actually allowed us to control some of the aspects of the recording robotically, so I could actually record on 4 workstations at the same time to increase my throughput. It was really exciting.
Well, thank you for sharing that story. I like hearing about the origin stories of all of our guests, because for our listeners, including many students, I think it's important to recognize when there's an opportunity that could speak to you that becomes your origin story. So thank you very much for sharing that. So you alluded to this briefly. Your primary education was in Germany, and now you're here in the United States, have been a professor for a long time. You built your own school of neuroscience. Tell us a little bit about the differences between the German educational system and what we offer here in the U.S.
I came 36 years ago to spend 2 years. Actually, I should have mentioned that after this ion channel work, I was so fascinated. I actually signed a contract with Bert Sakmann, who had then moved to Heidelberg. Still hasn't won the Nobel Prize yet at the time. But I traveled to a neuroscience meeting in Canada in 1987. And seeing just the sheer volume of science that was done in North America and the excitement that people had, I really had an urge to come to the States to do a postdoc. So I went back to Bert Sakmann and says, I've got a problem because, I have a chance to work in the best lab to do ion channel work in the world. Mm-hmm. But at the same time, I have this urge to spend some time abroad and experience, another culture doing science. His response was unequivocal. He says, Harry, absolutely. You should do that. I want you now. I want you then even more. You can come back in 2 years or whenever. He says, I did the same thing. You should definitely do a postdoc. So off I went to do a postdoc at Yale, and I joined a lab that was also studying glial cells. That's actually where some of the interest in glioma started, because I actually got entangled with a neurosurgeon there. And he cut out tissue either from glioma patients, from patients with mesiotemporal lobe epilepsy, where you take out the glial scar. So I was able to record from human glial cells that are still kept alive in a slice after its operation for 12 to 20 hours. And that was just really fascinating. It actually was one of the distinguishing features that kept me in the United States, the proximity between researchers and physicians. In a medical school, you basically coexist and you collaborate. There is hardly any of that. It certainly wasn't at the time. You've got the hospital on one side of the campus, and you've got the basic scientists on the other side. That is in Germany, they hardly ever talk to each other. And certainly this disease-focused collaboration on projects didn't exist. The other thing that didn't exist was a clear academic ladder where you could become independent with your own ideas, get your own funding, and start your own laboratory. At the time, more though than now, there was one professor and maybe an associate professor in each department, and everybody else was basically a staff scientist. Someone needed to vacate their position for you to be elevated to become the professor in the department. Versus here in the U.S., we're used to having 20, 30, some departments, 100 faculty members, ranging from assistant to associate to full professor, and they're independent at any level for as long as they obtain funding and build a research program. So I was really convinced that this is a much better system in which to thrive. And of course, the United States has an incredible funding system for science. And as much criticism as there is from time to time that it's hard to get grants, we actually have very generous awards given to us from NIH, NSF, the DOD, but also big foundations like the American Cancer Society, American Epilepsy Society, Cure Now, and the list goes on and on.
And presumably pharmaceutical partners, right?
So and it's really allows for fairly robust funding for many investigators to build these rigorous research programs. I think the sheer volume of funding that's being invested in science in the United States far eclipses the funding that occurs in any other nation, maybe except for China now. But it used to be certainly that we were at least tenfold above anything else, and that's another reason that kept me here for sure.
Well, thank you, Harald. We're glad that you've stayed. As a professor, for students interested in neuroscience at the University of Virginia, how can they get involved in your department? And also, is there some advice that you could offer to a STEM beginner who might be thinking of a career in neuroscience?
Well, I'm glad you asked. My department has 20 faculty members, and each of them has anywhere between 5 and 10 undergraduates that they give an opportunity to do research. There's over 100 scientists or professors on this campus that are happy to entertain undergraduates interested in doing lab work. And I would urge any student to look way beyond that, to just look at any basic science department, because at the end of the day, it really doesn't matter where you learn certain techniques. We all share similar approaches to cell biology, biochemistry, genetics, molecular genetics development. You simply have to find an environment that you enjoy showing up every day, where you have a good relationship to the mentor, where you feel you get something out, you really want to be there. Remember, you're not, as a student, you're not going to work on what you work on as an undergraduate 20 years from now in all likelihood. So it's all about instilling the science bug, figuring out what interests you. And, you might be a neuroscience student or thinking you're a neuroscience student, and you work in a cancer lab, and all of a sudden you fall in love with a different discipline altogether. So there's rich opportunities, and I think that the most important recommendations I would give them is If someone says, no, I don't have room for you, don't take it personal. They probably don't, because just like my lab often has 10, 15 people, keep knocking at the next door, the next door, the next door. Send out emails. Eventually, someone will be happy to have you. And once you have a little more experience, you'll actually become quite a commodity. Opportunities present themselves almost randomly, but they favor the prepared mind. So if you hold your mind open to see beauty and opportunities that present themselves, I think should really guide your path.
So we have a tradition here on Who's in STEM. I'd like to finish our episodes with a fun, unrelated fun fact. Can you share a tidbit, maybe from your childhood, maybe from your early education, or perhaps here in Charlottesville? Share with us a fun fact of your choice.
Well, fun fact, I ride a motorcycle for fun. Triumph, Bonneville, and I don't go fast, and I don't go on the highway. And you find me on a nice spring or fall day riding down, of course, with a helmet and protective gear, down the Blue Ridge Park.
Too.
Absolutely, and enjoying that. I actually rode a motorcycle through the desert of Australia when I was 22 years old, all the way from the very south of Melbourne through Ayers Rock up to Cape Tribulations. So that was a fun fact. But I do want to mention that my real passion is not motorcycle riding, but I think it's probably my kids, and I'm very proud in what they've been doing. Unfortunately, I haven't been able to pass the science bug onto either of them. But my oldest daughter, Melanie, she got a master's degree in German literature, and she's now very happily an academic advisor at Virginia Tech. My daughter, Sylvie, just graduated from her residency program in emergency medicine at BU. Mm-hmm. She moved to Arizona to join the Navajo Nation as a resident physician on a reservation. How can you not be proud of that? And then I have two younger ones, Charlotte and Carter, who are still in school and trying to figure out how to balance their athletics with academic work. And so there's still a chance that one of them might actually catch the science bug before too long.
So, Harald, this was a wonderful conversation. Thanks for taking the time to be with us today. I'm Ken Ono, STEM advisor to the Provost and the Marvin Rosenblum Professor of Mathematics, and you've been listening to Who's in STEM.
Who's in STEM is a production of WTJU 91.1 FM and the Office of the Provost at the University of Virginia. Who's in STEM is produced by Catherine Kosseboom, Claire Curzon, Benjamin Larson, Katie Nichols, and Ria Verma. Our music is composed and performed by Robert Schneider and John Ferguson of Apples in Stereo. Follow us on Facebook, Instagram, and Twitter. Listen and subscribe to Who's in STEM on Apple Podcasts, Spotify, or wherever you get your podcasts. We'll be back soon with another conversation about scientific and technological innovation at the university.